So these images that you see are what you would look at if you were viewing them under the microscope after employing the Gram stain technique. Now, there's a structure that is said that some bacteria have, not all bacteria have this, which is referred to as a slime layer or a capsule. And the proper scientific name for these structures is called the glycocalyx. The glycocalyx consists of gelatinous material that's secreted by the cell membrane and then built on the outside of the cell wall. It's very slimy and very sticky, which is why it's referred to as a glycocalyx, because it's basically a covering that is comprised of polysaccharides. There's two types. You have a slime layer and you have a capsule. The slime layer does not have as much structure to it as the capsule does. The slime layer is very loose and it's affixed to the cell wall. The capsule is going to be more organized and more structured, and it's going to be firmly connected to the outside of the cell wall. Some organisms that are pathogenic that express these glycocalyxes are referred to as the genre of bacteria known as Pseudomonas. Pseudomonas aeruginosa is an organism that comes to mind. It's an organism that you can find in a hot tub, which can cause infection, and it will contain a slime layer that surrounds it. And the slime layer enables it to adhere to surfaces as well as to protect itself from being phagocytized or being eaten by immunity cells. Organisms that have a capsule to them will be things like Klebsiella pneumoniae, which can actually be a lethal organism if you're infected with it. Neisseria meningitis, which can also pose a deadly infection to those infected with it. And then Streptococcus pneumoniae, which causes pneumonia. All of those possess a capsule and makes the bacteria very hard to be killed by your body's natural immune defenses. Because of the capsule, these bacteria are not able to be phagocytized by your immunity cells, and if they're not phagocytized, then they're not broken down, and then they're able to grow and develop inside the body and induce infection, which is what makes them pretty dangerous. So for us to be able to know if a bacterial organism expresses a capsule or a glycocalyx, we have to employ a new type of staining technique. The Gram stain technique is only going to be able to stain for peptidoglycan structures or a lack of. But if we want to figure out whether or not a glycocalyx is present, we have to do something that's called a capsule stain. And the capsule stain is kind of an example of what we refer to as a negative staining technique, because we don't actually stain the bacteria. Well, we do use one reagent to stain the bacterial cell, but we don't stain the capsule. The capsule, as you can see in this image, is unstainable, just like it's not digestible, or it makes the bacteria unedible from phagocytes. So in this particular example of a stain, you have essentially stain being added, such as Congo red, and that Congo red is added to the background of a slide. And then you have another stain that's used to stain the actual cell or that's surrounded by the capsule, but the capsule itself is not stained, and so you get this kind of halo effect. And if you see this halo surrounding the bacteria cell that's underneath the capsule, then you know that you have the presence of a capsule. So this is what we refer to as a capsule stain. So I told you we were going to go back to the structure of the flagella. Prokaryotic cells are some will have a flagella, some will not. Most bacterial cells that have a flagella are a rod-shaped bacteria called a Bacillus bacteria. The flagella provides motility for the prokaryotic cell, and they can exist as either a one flagella that exist on the outside of the cell. So if that happens, then you have what's called a monotrichous bacteria, where you have a single flagella that's used to propel the bacteria. Some bacteria have two flagella. You can have one bacteria at both ends of the bacterial cell, and if you have that, then we refer to that as an aphetrichous bacteria. Bacteria that have flagella at one end of a cell, and this can be more than one flagella located at one end of a cell, we call that Lophotrichus. If you have bacteria that have flagella all over the cell, such as a Salmonella bacterial cell, we refer to that as Peritrichus. The number of flagella that a bacterial organism has is dependent on the species. So this is another structure that we use to help us identification of what type of bacteria might be inducing infection. And all of the flagella are comprised of the same structures, which are referred to as flagellin, which is why they get the name flagella. And flagellin is just a simple protein used to build the structure that the bacteria use for motility. And here's some different images of the different groups of flagellated bacteria that we talked about. The Monotrichus just having one flagella, Aphitrichus having two flagella, one at each end. Lophotrichus is going to have multiple flagella at one end, looks kind of like a little beard. And then for Peritrichus bacteria, they have flagella situated all over the body. And here's a microscopic image of a Salmonella cell that has multiple flagella all over. So this one is going to be an example of the Peritrichus bacterium. Okay. The other structure we want to talk about is that of the pili. I mentioned this earlier before. Remember, the pili are used for adhesive properties, which help bacteria to adhere to different surfaces. The pili are very thin in structure. They're very hair-like. You typically see pili on gram-negative bacteria. They're made of a protein called pillin, hence why they're called pili. The pili are different from the flagella because they're thinner and they're typically shorter. They're not, again, used for motility purposes. They're used for adhering purposes. So they help the bacteria to affix themselves to a particular location. And as I said before, bacteria can use what's referred to as a sex pilus in the process of conjugation, where the bacteria is transferring genetic material from one cell to another cell. Here's an example of an organism that has pili on it. It's called Proteus vulgaris. You can see the pili structures that are situated on the outside of it. And then as a comparison, you can also see flagella on the outside of the cell. And remember how the flagella, they have different locations, either at polar ends of the cells, one end of the cell, all over the cell. But they differ from the pili because they're much longer, and the pili are much shorter and sturdier than the flagella, which are going to be more used again for motile purposes, while the pili are used for adhering purposes. Another structure that's important to know regarding bacterial organisms is called endospore. These are also referred to as simply spores. They are not spores in the sense of fungal spores or plant spores that are generated for the process of reproduction. Endospores are generated because they are a survival mode that bacterial cells will undergo when they have been depleted of certain resources. So when you think of an endospore, think survival, don't think reproduction, because they're not used for reproduction purposes. There's a few genera of bacteria that deploy these types of structures. The Bacillus genre and the Clostridium genre. I guess one organism you might be some of the organisms you might know of in these families are Bacillus anthracis, which is the organism that causes anthrax, or Clostridium botulinum, which is the organism that can cause botulism. So those particular organisms are very scary. They're among the most dangerous of all the bacteria, and we refer to them as a threat to our human existence because they produce these endospores, which we as humans can breathe in, and when we breathe them in, it causes us very deadly infections if not treated promptly. And that's all because of endospores. Endospores have very sturdy structures. They consist of these thick walls that help to preserve the genetic material inside of them. And once these endospores come into contact with water or an environment that's more conducive to living in, then they simply germinate. And when they germinate, they'll start to produce viable cells, which will initiate infection. So these genres that are able to generate these thick-walled endospores to survive, they do so through the process of sporulation, which again is not reproduction, it's sporulation, where you have the generation of an endospore. And these endospores can survive for thousands of years. They're resistant to heat, so they're resistant to extreme temperatures, they're resistant to cold, they can be preserved, they're resistant to drying, they're resistant to chemicals. So if a bacterium is producing endospores, it's really hard to eradicate endospores. We have to use specialized techniques to get rid of the bacteria. And one spore is generated inside of a traditional bacterial cell once a bacterial cell is depleted of nutrients. So what happens is the genetic material inside of the original bacterial cell is isolated off, a wall is formed around it, and then an endospore coat is formed around it. And so what happens is that once the endospore is generated, it will release itself from the original cell and it will just float off into the environment, leaving a vegetative bacterial cell behind. So in order for us to figure out if certain bacteria employ this technique to survive, we use a specialized stain that's called endospore stain, where you add a dye called malachite green and you use heat or steam to infuse that malachite green into the endospore so that way we can visualize it. And here's a nice image of what an endospore looks like. So remember, these are not reproductive structures, they're survival structures. So the endospore is generated here inside of this bacterial cell. So this bacterial cell has been subject to an environment where there hasn't been a lot of resources, and that's caused the formation of this endospore, which is basically a sectioning off of the genetic material and the development of a wall around the genetic material. So essentially this endospore is kind of like a little space shuttle that says, oh, we're packing up and we're leaving this bacterial cell and we don't have enough resources to survive here. So the endospore is generated and then eventually it's going to be released from the original cell, and then this original cell becomes what's known as a vegetative cell. Okay, so now we're just kind of highlight, the differences again between prokaryotic cells and eukaryotic cells, which are the main points that I want you to take away from this lecture. So we've already covered all of the structures that I think that you should be familiar with regarding bacterial cells. And as always, please email me with questions if you have any. But remember, eukaryotic cells have a true nucleus, which means there's a membrane that surrounds the nucleus. Prokaryotic cells do not have this. They simply have a nucleoid region where their chromosomes are pretty much free-floating in the cytoplasm. Eukaryotic cells are essentially divided into two types. We have plant cells and we have animal cells, and that's pretty much it. The plant cells contain a cell wall, and the animal cells do not have a cell wall. Eukaryotic cells have organelles to them, which are membrane bound, and all of those organelles have specialized structures. Prokaryotic cells don't have any membrane bound organelles, and the only membrane that you'll find in a prokaryotic cell is going to be that of the cell membrane that surrounds the cytoplasm. Okay, we haven't really talked too much about this, but we're going to talk about kind of the reproduction of organisms as well as how their cells reproduce. In prokaryotic cells, remember I did mention this earlier, you have binary fission, which is basically one cell splitting itself into two daughter cells. And when it does this, it generates two identical daughter cells. So the genetic material in both of those cells are exactly the same. And the reason for this is because before the cells before a prokaryotic cell divides, it's going to replicate its chromosome. Once the chromosome is replicated, it'll create a wall that will section off the two cells and then generate two daughter cells that are identical to each other. And when this takes place, the first time, we just call this a simple generation time.